Okay, perfect. Hi everyone. So my name is Suyenna, and today I'll be presenting on organoids, and specifically organoids and their application in the drug discovery process. So first, a bit about me. I'm from the next province over, from Saskatchewan, and I'll be entering my final year studying biology with a minor in philosophy. And I joined the Wishart Lab beginning of last year as part of the science internship program. So in today's presentation, I'll be covering 3 topics. The first just being a brief description of the drug discovery process, and then second about explaining what organoids are and addressing how they can deal with the limitations in the drug discovery process. And then finally, I'll conclude with a pretty cool example, I think, of organoids being used in drug discovery. So first, just a very quick brief note. This is a very broad and general overview of the drug discovery process, and I'm by no means an expert on this process. It's just to give some context of what organoids can do. So first, the drug discovery process is divided into 4 stages. In the first 2 stages, the early drug discovery process and the preclinical phase, the goal is to identify a potential disease target. So this could be a protein, a gene, or maybe even an RNA sequence, and put possible compounds for that target. And then here, multiple tests will be run to validate and optimize the compound to gather information about its safety and efficacy. In the third stage, which is your clinical phases, it typically has 4 phases, phase 1, 2, 4. And each phase have increasingly larger cohorts of individuals who are being tested with the compound. And it also examines safety and efficacy, as well as dosage and if the compound has any potential side effects. And then finally, the last stage being regulatory approval is where all the data that needs to be collected, analyzed, will be collected and analyzed and then submitted to the appropriate authorities for review. And approval is necessary before the compound can be sold and enter the market. So, so I'll be looking specifically at the application of organoids, mostly pertaining to the early drug discovery process. So a bit about the early drug discovery process is where you identify potential targets, and also identify the compound specific to that target. You can do various types of screens to identify the hit compound from the thousands of potential candidates. And you also do many secondary assays like cell-based assays and also biochemical assays to characterize the efficacy and its safety profile. However, even with meticulous and careful work, there is still a large gap between the success of identifying the compound and also for this compound to successfully pass all 4 phases of the clinical trials. So some of these limitations of the early drug discovery process includes a discrepancy that can exist between your in vitro assays and also the complexity of the pathology in vivo. So for example, if you do like a cell-based assay to examine the effects of the compound on a particular cell line in a petri dish, well, the cells grown on the dish is not a good reflection of what the cells look like or how they'll be organized or how they respond to the drug in vivo. As well, there's limitations with using animal models. Not only are there ethical issues that should be considered, but animal models are also not a good reflection of what can occur in humans. And finally, there can be a lengthy timeline during the early drug discovery process. It can take up to 3 to 5 years to identify a promising drug candidate. So moving on to a bit about what are organoids. So organoids are in vitro 3D cultured miniatures of organs that originate from stem cells. So you can think of organoids as these like mini clusters of different cell types that come together to organize themselves to have similar architecture and similar morphology to the cells that you find in an organ. And then these cells can then replicate some of that organ's functions. So on the right, there's an image of a neural organoid, and it's showing immunohistochemistry. So when they do an immunohistochemistry analysis, they'll stain for markers or molecules on your cells that are indicative of the cell types that you expect to see in that organ. And what's really, really cool about this image is that they stain for CTIP2 in green and LHX2 in red. And these markers show that the organoid was successful because you can see the different layers of the cerebral cortex. So I just want to dive a bit more into explaining what 3D cell culture is and why is it so important compared to the traditional 2D culture. So if you see in the left image, that's your traditional 2D cell culture where your cells grow on a monolayer in a flask or a petri dish. And because they're grown on a monolayer, there's a lot of implications for the cell's physiology. The cells are forced into a planar shape, which doesn't necessarily always mimic what you see and their native structures in vivo. And furthermore, the 2D cells, because they're grown in this monolayer, they're deprived of like cell-to-cell interactions and also cell to the extracellular environment interactions. So then there's also a lack of microenvironments and niches. Whereas you compare it to in vivo, like in your 3D cell culture in the image on the right, you can see that the cells are grown in all three dimensions, and they can also interact with each other and also interact with their extracellular environment. So you see that some cells will be within the aggregate and some cells will be on the outskirts. So if you recall some of the limitations of the early drug discovery process, the first being the complexity of in vivo pathologies. Well, instead of using like your traditional 2D cell culture on the left, if you do your testing in 3D cells whose morphology and physiology is closer to in vivo conditions, then you can also, your drug penetration will also be taking into consideration like the architecture of the cells. Unlike 2D cultures, where all the cells are exposed to the same amount of nutrients and toxins. As well, 3D cells can also address differences in animal models because the 3D cells are grown from human stem cells, so they'll more closely respond to the compound of how you see like your body. As well with the lengthy timeline, if you identify your drug candidate, the timeline to identify the drug candidate can be cut down because if you're screening in an environment that's, again, closely more mimicking to in vivo conditions, like say an organoid, then you'll be able to notice any problems with safety or toxicity earlier. And then, so finally, I want to just show this really cool study that's looking at liver organoids and drug-related hepatotoxicity. So a recently published study was looking at liver organoids as a possible method to accurately predict the risk of DILI before the drugs are sent for clinical trials. So DILI is drug-induced liver injury, which is a result of an acute or chronic response to a compound. And it can lead to acute or chronic liver disease. And there's typically two mechanisms that can cause DILI. The first being intrinsic, which has a predictable mechanism. The risk of getting DILI is reproducible based on the drug dosage, and also if the drug's already known to cause liver injury. However, the second mechanism, idiosyncratic, is a big problem for clinical trials because the mechanism is not predictable and it's also not reproducible. It can occur in patients independent of the dosage given, and the exact mechanism is also not known. So the risk for idiosyncratic DILI is very hard to predict, which can make it a major problem for successful clinical trials of identified drug candidates. So recently, there were clinical trials which were run for a drug called anagrivir, which treats hepatitis B. And when it was tested by itself, the drug displayed no signs of toxicity. However, in a later phase 2 trial, when anarigovir was tested in combination with another drug called tenofovir. Some of the patients displayed symptoms of severe DILI. So this showed that there was a great need for to find a preclinical method to predict the risk of DILI with high fidelity, especially since idiosyncratic DILI is so hard to foresee. So what the authors did is that they grew liver organoids from induced pluripotent stem cells. And then from those organoids, they used two different models to see whether they were accurate reflections of the liver organs. So what happens is that for the first model, they dispensed the organoids into a multi-well plate for high contact screening of the different compounds. In the second model, they put the organoids on a microfluidic chip, which is also called an organ-on-chip. And in brief, this chip has very small channels where you can put matrix, you can flow also nutrients, oxygen, and media. And you can also control the rate of which these components are being flowed. So the organoids are put in an environment that closely mimics what you see in vivo. And then both of these models, the organoids are exposed to the same set of compounds as the ones on the plate. So I just want to highlight a couple of the results that they found. First, the drug-treated organoids displayed DILI biomarkers reflective of what you see of hepatocytes in vivo. So they noticed that there was decreased albumin production, which is a sign of liver toxicity, as well as there was elevated levels of two enzymes, alanine aminotransferase and aspartate aminotransferase, which are also signs of liver toxicity. And what was really, really cool is that they saw that the organoids were able to differentiate between the monotherapy of just anarigovir by itself, and also the toxicity that was shown in the combination therapy of anarigovir with tenofovir. So as a result, the study showed that it's really important to use organoids as the in vitro preclinical drug testing model because they can provide a better indication of what can happen in vivo. And that's it for my presentation. These are my references. And as well, I also want to say a huge thank you to Dr. Wishart and Prashanthi, and also to my direct supervisor and amazing mentor, Upasana, and the rest of the members of the protein team. So yeah, thank you. And if anyone has any questions, I will do my best to answer them.